let them all in. Hello, everybody. Welcome. This is week 5, LS23L. Let me bring up my PowerPoint. I don't see any student. I just see icons. Oh, hi, David. Hope you're doing good. Let me share my screen. All right. So I see one student and I'm sharing. Oh, PowerPoint is here. Okay. All right. So I'm all set. So hello, everybody. We're almost at the end. You have one more lab next week, and then you have your video presentation. And we're also going to start your last writing next week. And let's see, we have 2 big labs this week. One is PCR, and PCR is kind of complicated to understand. So students sometimes don't understand it right away. So I have many slides for this, and I might just rush through, but to just get you started for your section. And then histology is historically also a lab which people are struggling with if they have not taken LS7C, or they're not really so familiar with microtissue and microtissue microscopy. So it's also a difficult lab. So we have 2 difficult labs this week. But they're also exciting, PCR and ELISA about SARS-CoV-2. And then a couple of work slides about your group work, because your video is due next week, Monday. So again, that's me. I just watched the Black Widow, which is an interesting take on action movie. And so I just wanted to quickly check in with everybody, how everybody's doing. I did that during the SARS-CoV-2 to check in with everybody. Everybody's in the vacation mode at this point. So I put it up here. No, I'll wait a little bit. Everybody, is it working? Doesn't seem to be working. There, now they're coming in. Also, I think I forgot my first slide. So Rachel and Brittany are here. You can type in the chat what worked for you the last 3 weeks since we didn't see you, and what is working. And Rachel and Brittany will answer your questions. And then we also can later post the Q&A online. So now I'm getting some feedback, and somebody already texted in chat.
Yeah, I think this is their first time doing the Menti poll, maybe. I think we skipped it maybe in the first week, so might feel a little new. Okay,
so, but it is working. So there is a code, a website and a code you can go to. And so people are tired. Yeah, we're towards the end, and this is a marathon, as you probably realized, 18 hours a week for my class only. You probably have other classes too. Can't wait to be done. Yeah, yay, 2 more weeks. This is week 5, no, 4, right? 4, yeah. Okay, well, thank you. Doesn't look too bad. Most people are happy. So I guess the Corona crisis right now is a little, moved on for now. We might not be done with it yet, who knows? So, okay. All right, so then if you need help, if you're really stressed, again, here's some resources for you. Campus Wire, most of you are on Campus Wire at this point. So again, there's one more writing we start next week, so utilize it. Before tomorrow or Wednesday, depending on when your section starts, you have a group slide due for PCR and ELISA. And there's 2 labsters, one for PCR, one ELISA, and it's 110 minutes on average. So, and then there's a couple of movies you need to watch. So make sure you have enough time before you go to your section. So here's the slide. How are PCR and ELISA tests used to detect SARS-CoV-2 and COVID-19? So if you already did that slide, you're a little bit prepared. And I have a couple of slides also today about that. Both labs tests are due before your section, so make sure you have enough time. All right, then scores as usual, they're gonna be updated in real time on my UCLA. Everything should be updated at this point. Group slides, worksheets will be for last week will be updated by Wednesday. Also the asynchronous worksheet after Tuesday. And the practicing peer review from last week will also be updated. So then all grades are up to date, and then please check if grades are missing and let us know. So report 2, you just submitted your report 2. We're still waiting for the late submitters till today at 5. And then this week, you're gonna do the assessments and the optional reflection. So you did this. We are right now here. Tuesday, tomorrow, we start the peer reviews, the assessments, and then at the end of the week, like the first time till next week, Monday, you have the optional reflections. There is a late submission. Due was last Sunday, yesterday, 5 p.m., but we accept till Monday, 5 p.m. So there's a couple of stragglers. So we are in week 2 of the writing assignment. So here's the assessment. Let's see what else. If you're late, you get 5 points deducted per day. This deadline on Friday at 5 p.m. is a hard deadline. If you miss the assessment, you lose 25 points, right? So this is a higher stake. It's 50 points total, 25 for the text and 25 for the peer evaluation. What else can I? That's it. That's all I can say here. And then again, you get 6 plus your own. So don't forget to do your own. And also leave yourself enough time to evaluate it using the rubric, right? The rubric. And then if you haven't done the PPR, and that's a slide, there is a for review, not for points. If you miss this, you miss 5 points, but you can review the rubric and the evaluations here before you evaluate your peers. So make sure you do that, so you're on board with everybody and you get a good evaluation score. And then we check again for plagiarism. Most of you at this point have done the tutorial. The ones who got a 0 in the first one, you know, it's like, oh, it's not me. But if you get a score in your second one, you get a warning, and then at some point I have to report you. So if it's above 40, again, I will be meeting with you. So just like in the first round. There's no warning for the third one. In the third one, if you have a match, I will have to report you. All right, and then for the second one, what do I check? The title, the hypothesis, the results, the discussion, and the citations. Now, if there's a match here and there, 2 different papers, that's fine. But if you match even at 10%, a little bit here throughout the whole paper, I know you have taken another paper as a blueprint. So that could be a problem. All right, so the optional reflections are open on Saturday. They are due on Monday next week, 5 p.m. So it's a weekend work you have to do here if you want the 5 extra points. All right, deadlines. There's 2 labs for this week. So it's the Tuesday, Wednesday lab. It's lab 7, which is PCR and ELISA. And then we have histology. So this is, and then you try to work from left to right again. Deadlines, the group discussion slide is submitted here before your section, the labs are before your section. And then here's a lab manual and the videos. And then here's a lab activity in section and asynchronously if you're not going to lab section. And then here's what you have to finish for writing. There's a group presentation tab for next week for the video presentation and the post lab. Deadlines as usual, pre-lab, labs, the group slide are due before your section. So whenever your section is, that's before. And the same for the second lab. Next week, you're going to do the reflections. This week, you're going to do the assessment. They do by Friday, 5 p.m. Last week was do the practice and peer review the text. So that's roughly, but then if that's too complicated, again, In the deadline tab on each lab, you can just click on these and go directly to the assignments, and they're listed here and here. All right, so let's go to the first lab. So again, it's a pretty big lab. It should have been covered in 7A, that's molecular biology material. And students do get confused. It's not an easy concept. So we try to provide you first with the Labster. So that's 40 minutes for the PCR, 70 minutes for the ELISA. Here's a background PCR video from me, which also goes into how it was invented and who discovered it. And then this is more about the technique. Wait, this is more about the technique, but also there's an addition for SARS-CoV-2. You need reverse transcriptase because SARS-CoV-2 has mRNA, not DNA, and PCR is based on DNA. So we have to convert the mRNA to DNA first to test for the virus. So this is about testing for the virus. This is just general PCR. There's an animation for general PCR to just understand what happens. And you're doing primer design, so to understand what the primers are doing. And then there's two for SARS-CoV-2 testing lab, how the samples get processed, how fast it goes. So there's two little videos here. And then there's another background video on ELISA, which is also used for SARS-CoV-2 testing. The video is a total of 55 minutes and then 110 minutes. So make sure you have to do all that before you're going into your section. So again, labs is about PCR, and there's how it's used in forensic DNA profiling to make it interesting. And then ELISA is also about proteins and how to quantify proteins. So there's also a case and in a lab environment. All right, then in this section, there's going to be the question, share a little bit about yourself if you feel comfortable. What's one good thing that happened to you in this week? So think about that. I don't have an example right now. For me, so in lab, so basically we're introducing PCR, and the first thing you're going to investigate how PCR is used for COVID-19 testing. And then you get the sequence back you used already last week. Remember, you got the viral sequence to look at changes in the sequence. You got a primer pair. You have to look for the primer sites in the sequence, and then look at the mutations at that site and how would that affect the PCR. You're doing the same asynchronously from home. There's a worksheet. So here's the SARS-CoV-2 genome. Again, it's mRNA. And you're going to look at the changes in specific regions. And then when you want to basically design primers for a specific sequence, there's these little where you design it, and then you see how it works in silico. And then you run the wet lab and you see how much product you got, PCR product, and then you can refine it. And you do a mini version by evaluating how good are your primer pairs. And to do that, you have to understand PCR, you have to understand how to read a sequence, and so on. There's also in one of the lectures, what are good primer pairs? It's also in the lab manual. If you want to design a primer pair, there's specifications here. You can read through this and then decide with your primer pairs, do you have a good primer pair or they're problematic? So then when you go in section this week, the first breakout room, you get assigned a primer pair, and then you try to locate these in the SARS-CoV-2 sequence. There's also a quiz at some point, I think. Is that still there, Rachel, Brittany? Yeah, so there's a pub quiz at some point, how to understand how the primers bind. So if you're not prepared, you won't be able to know what the answers is to that. And then in the breakout room session 2, you're going to evaluate your primer pair. And then you report back. So this is the questions, which the TAs will select one of yours and present it to the group. So basically, we extended the PCR to RT, reverse transcript PCR, and that's one of my lectures. It's also qualitative RT-PCR, and there's a confusion about that. That's also in the lecture. But the viral RNA tests, there are, so that's a PCR, they're more reliable because it's so specific than the protein, the ELISA, right? So there's some rapid tests. You might know a little bit because we went through SARS-CoV-2. So try to think about this. And then there's also, you can test the immune system. And that's still under investigation. And then, so here's when you do the ELISA, and then here at the beginning, you do the PCR, when you can actually look for the virus itself, for the mRNA of the virus. And then the same here. And so you get an infection, you are exposed. When can you see the virus? When can you see the reaction of your body, the antibodies? So, so that's what this lab is about. And if you prepared the slide, you know a little bit about it. So here's the sequence, and you looked at this last week. There's a notation here. These are mutations, like +9 means there's more than or about 9 mutations on this position. The other have some mutations, 1 to 5 mutations. And then these are the primers. There's a forward primer, the reverse primer, and this is the probe. So you're gonna get data like that, and you have to search the sequence where it would bind. And it's very specific binding. Then you're also going to analyze your primers with primer 3, which is a standard program used bioinformatics in labs. You know, how good are the primer in theory? These are theoretical. And then once they are in theory looks good, then you can go into the lab and test them out. All right, and then there's a pop quiz. Where does what bind? And so breakout room session 1, locating the primer pair. And then there's a worksheet with 15 questions relating to the primers. So again, if you're not prepared, you have no idea what's going on. So be able, you know, to answer these questions. You need to know what the forward primer is, the reverse primer, what the probe does. And then here's an example. So you get a forward primer, reverse primer, and probe. And there's a 5'3' orientation, which is important here. And if you don't know what that means, you watch the lecture, and then you have to flip the reverse primer to read it in this direction. So it's AG here, TA, and then it's AT backwards. So that's a flipped one. And that's important. So it's not nonsense. So if you don't know PCR very well, you're going to just be confused. Now, how long are the primers? You have a start position, a stop position. And so how do you get from the stop to the length? So there's going to be a question like that. So, and I have another slide for that. So this is an example how the primers bind on the sequence. Now, the length, so you have a start and a stop position here. It would be 87 minus 84. So that's 3, but the primer is actually 1, 2, 3 positions, 4, right? So you have to 87 minus 84 plus 1, and then you get the number of nucleotides. So that becomes important again once you do the worksheet. And then this slide again, how good are your primers? And that's based on primer 3, and then you can see how long are the primers, what is the GZ content, the melting temperature, etc. That's breakout room session 2, where you're actually going to be evaluating your primer given the criteria. And so here's an example. So there's some mutations in the primer region here, and there's one larger mutation here. And then here is, will these mutations cause the primer, disrupt the primer binding? So you're going to analyze your set of primers. And the sequence you're getting is actually a live sequence from SARS-CoV-2. And so these peaks might look totally different last quarter than they look right now, because it's mutating, the virus is mutating. And then you try to think, right, after the virus mutated, do the primers still bind? Does the PCR test work still? It might not work anymore after a while if there's too many mutations. And then you can look at the geographical distribution of these mutations of your primer site. Will the PCR test still work, right? So it would still work in South America, mostly in North America, but then there's a single country here where there's a significant amount with mutations where the primers might not work anymore. So you're gonna think about that. And then you have more questions to answer. So it's a long questionnaire about your primers. And you can't just wing it. And then, so here's a primer 3 output with the information, the sequence, and the melting temperatures here, GC%. And again, you want to answer then for your specific primers, you have to run primer 3 and then analyze this data and able to read this data too. And then for the asynchronous activity, you're doing the same thing. Just you get your worksheet, you get a primer pair and a probe assigned to you, and then you go to Google Sheet and you start working through the worksheet. And that's due Friday by 5 p.m., Sunday by 5 p.m., sorry. Okay, so that's it for lab 1, PCR. And then we're gonna switch gears. We're gonna go into histology. And that's a physiology lab, traditionally going along with 7C material. So you have a group slide too, and this is a group slide. What would you expect to see when looking at the cross-sectional slide for skeletal muscle and why? And then how might the longitudinal section look different? So if you not have had 7C, that's going to be a difficult lab for you because it's about tissue and micro tissue, how our nerve, the brain, how its composition is, and the muscle cells and fat cells, and organ tissue, you know, heart cells or kidney cells. You know, how does that look in a microscopic, if you look at the kidney, for instance, and then how does that look under the microscope? And so this lab is built on that you think about the function of the organ and then kind of have a guess what it might look like under a microscope. So, so that's what the histology lab is about, and I have a couple of slides for that. So, so you want to submit your slide before, that's Thursday, Friday, right? You go into lab 8. The question in the section is, in your opinion, what's the most beautiful place on earth? These are my COVID-19 walks at the beach in Santa Monica, and it was really beautiful. It's a little more populated now, but we were pretty lucky to have this available during COVID-19. So this lab also introduces microscopy, and so labs that's doing the heavy lifting here, having a simulated microscope to use. There's also a tutorial which walks you through 2 different microscopes used in the lab traditionally. There's a background lecture on histology. So this is a tissue. This is an egg cell, actually. This is the ovary. And I can just know that because I've done it so much. You know, if you've never done it, you look at this, you have no idea what it is. So in the tutorial, it walks you through a couple of tissues like artery, blood, bone, liver, lung, brain, skin, and muscle tissue, and how, what the function is of these organs, because then from the functions, you can maybe propose what it might look like under the microscope. Then if you're working asynchronously, there is a quiz you're going to do online. It looks similar like the tutorial. What else can I tell you? So maybe to prepare for your section, you want to do the lecture, you want to watch the background lecture, do the tutorial with also tips and tricks of staining. There's different ways to stain, and also in the background lecture goes over the staining techniques and how it makes different tissue look different and why. And then the tutorial is going over the function. Okay, yeah, that's it. So then that's already what I said. So we're trying to design the lab. Basically, you know the function of the organ and what it kind of does, and then filter blood, or you know, the heart is pumping. It's a mechanical tearing could happen. So how would you build an organ to withstand the tearing of the heart, right? And maybe that can give you a hint how it would look like under the microscope, right? So these are the tissues we're looking at. And then there's staining. So for some of the stains, the nuclei stain dark purple. So if you have a tissue with very dense nuclei and not a lot of cytoplasm, they usually look darker. then a thing where you have a little bit of cytoplasm and staining, so they look pinkish, or where you have no nuclei and then it just is red, right? So I, of course, familiar with that. I know what all these are, but if you are seeing this for the first time, you have to think your way through, why is this dark? Why is this lighter? And why is this really bright red? And there's a reason for that. And it has to do with the function. Here would be one example of one organ, the adrenal cortex, where it's darkly stained here, the less so here, it's lighter here, and then another intermediate color here. So there's 3 different layers of tissue within one organ. And there's a reason for that too. Labs that will introduce light microscopy and fluorescent microscopy. If you go in a lab, like a physiology lab like that, these are really helpful to introduce you to once you go into a lab in upper division, so you're familiar with microscopy. So, so then the goal of this section is making connection between morphology and function, what I just said, right? So the heart, how does it look, microscope, and what's its function, and why is it built a certain way? And then how would it look under the microscope, basically? And so you're gonna do an assignment for that. So that's a student question. And again, the TA will present one of the student slides. So in breakout room 1, you get assigned a tissue, a known tissue. So you get, say, like, here, this is your tissue. And then you put together a presentation with your group. And I have a couple of examples for that. And then you report back. Everybody is reporting on their tissue. So you get 8 mini presentation on 8 different tissues, how it looked under the microscope and what the landmarks are. And then you're going back into a room and you get an unknown pathology slide. Now you already have seen 8 different tissue, and the pathology slide might look similar to what, so you have to actually pay attention here, because you're only gonna work yourself on one tissue. So then you have to pay attention to the other group's tissue, because you get that unknown pathology slide and have to identify it. Then you come back out and maybe there's going to be a discussion, because both of you think it's, you know, some tissue, and it's not. There's only one unique tissue. So then that's going to be a good discussion, and it's okay to be wrong, right? So that's where you learn. For this activity, we found this great website, and it's free. We're nervous that it's free. And every quarter that it's going to be discontinued, but we communicated with people, and it is for education. It's a wonderful site. Rachel found it, and so, but then here's the link to it. So if you just go into section, you might not know how to navigate it. So if you just come in prepared, and you lose a lot of time just knowing the website. So I would recommend, here's a link, go to the website and pucker around, you know, click here and there and figure out, because you have, it's like a microscope. You can zoom in, have different magnification. And, you know, it's like, it's a tool, and you have to get used to the tool. It's like the Next Train website. So if you just think you can just use it, you know, maybe it takes you 5-10 minutes to figure out. So do it before you go to section, you're pro, and then you can faster answer the questions you actually have to answer. So if you totally crashed with the Next Train website in lab last week, you know, remember that and come prepared. So basically that's the example. So the TA will go over. So say you get assigned to spleen, and they're like, well, look up the spleen, right? So then you put together a slide. So you find something online. Where's the spleen? You know, this is the spleen, what it looks like, what's its function, and then what would it look like under the microscope, right? And then you figure that out with this website, basically. And then you can also, well, why does it look? You know, then you see what it looks like, and then you have to think, well, it's a spleen, it's doing this and this, it filters blood, right? And why does it? And then this would be the histology. So how do you get from how it looks here to what it looks here, right? And then so students pointed these out, these structures, and why are these and what do these structures do? And then once you know that, and you would see something like that under microscope, it's like, oh yeah, this is spleen, totally. right? So once you're experienced, you totally see it's clean. But like if you're not, you have no idea what you're looking at. But so that's what we're trying to teach you. It's not like blind learning. There's a reason why it looks like that. And that's our point, you know, how to make a connection from here and its functions. Why does it look like that? So everyone gets an organ and basically a tissue, and then you make you put together two slides, right? Like an overview slide like that, and then what histology, and there could be different magnifications too, if it helps, right? This is just one magnification here. All right, and then you're going back in the breakout room, you get an unknown pathology slides, and then these are the ones we have. And you just heard a whole presentation by all of your peers. How does healthy tissue lung look like? How does healthy arteries look like? Healthy liver, healthy skin, neurons, bone, blood, and skin, right? So how does that look in the healthy? So that's what you usually look at. And then you get this unknown slide, and then you have to figure out what is this unknown slide. And then you can discuss that with your group and do some research. And you know, and then you come back with a hypothesis. I think this is, you know, neurolemon. Okay, I can't even say that word. This is melanoma in human skin, right? So you say, okay, I think this is skin because we saw that skin before and had three layers, and I think I see the 3 layers, blah, 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 right? And then maybe somebody else said, no, I think we have that, right? So then you can discuss till you all figure out the right answers and then learn along the way. So that's how we set up the lab. Now, if asynchronously, you unfortunately don't have a sparring partner to figure this out, but what you get is 8 different tissue and presented, and then you have to figure out what it is, and then annotate what's the function and why does it look the way it looks. So you get 8 tissues, they're randomly assigned. There's 3 different magnifications, so you can zoom in and out, and you review the images, right? And then you by exclusion, right? What's easier to, if you know something else is this, and then you just go around till you get stuck maybe at 2 slides where you don't know, and then you can go into detail on that, right? That's what it looks like. Here's the identifications on CCLE. And then on CCLE, so this is a tutorial. So you can run the tutorial side by side with CCLE. So here's CCLE, and it's going to ask you a bunch of questions. But then if you have a big enough screen, you can open the tutorial here, where there's also a link in CCLE, and you can work your way through these examples and then answer the question in CCLE. Right, and then also in CCLE, there's also the images. So this will present the images. If you don't want to use this at all, you can do it first and then go to CCLE. And then if you click on these, you will see the images, and then you know, start investigating what it could be. All right. Wow, I'm really fast here. So then, and then we're done with lab 7 and 8. And then next week, we're doing human physiology. It's also a 7A lab with physiology, and you're gonna submit a group slide for that. What is the fight or flight response? Physiologically, what is happening in the body when this occurs? And you wanna submit that. And though this lab is gonna introduce your last writing assignment. So historically, we collected a lot of data with students. And again, there's going to be a database, and there's a lot of more experiments, way more experiments than what we did for the goldfish lab, right? The goldfish lab was kind of limited, but there's going to be a lot of things you can do, and it's also full paper. So you write a title, introduction, material, methods, results, and discussion. It's going to be a full paper, and hopefully you're an expert writer at this point. It's worth 80 points. It's a heavy load. But has a lot of cool experiments you can do. And so we're going to introduce that next week. But so this is to get you started to think about physiology. All right, and then the group video. So we introduced SARS-CoV-2 in week 3, right? That was last week. You did you looked at the RNA sequence of the virus. For the spike protein, you looked at the mutations on the nucleotide level and the protein level, how it might affect the spike protein. Now this week, you're gonna look at the sequence again, but in terms of PCR, where does the primer bind? And are there mutations at this position? Will it affect the PCR, right? So that's what you're gonna doing this week. You're also gonna look at ELISA, which is also used for testing, where you look for antibodies, and you learn a little bit about that. So then basically you already we have introduced SARS-CoV-2, COVID-19, the spike proteins, the proteins of SARS-CoV-2, how the sequence looks, the underlying sequence. And now you can go into basic for your video. You have one more week left. Video is due next week, Monday, 5 p.m. Make a small video, right? 8 minutes about the vaccination. And you can give backgrounds of the virus, what type of virus is, and I guess we have a whole list here of also how was the vaccine developed, what type of the vaccine it, what phase is the study in, how is the vaccine produced. You can give a little background on COVID-19, SARS-CoV-2, and then have a presentation about the vaccine you're assigned to. So it's an example video here from our very first quarter when we did this. This example is actually about one of the proteins. It's not about the vaccine, but if you look at this, there's 3 students, they're all presenting. They could be on video or not on video. You could just be in the background and narrate, but everybody has to participate. You can't not participate. Otherwise, you're not eligible to get points for the group presentation. And then Rachel put together some instructions how to answer all these questions. How to search online, how to do the research, how to access PubMed and websites. And it's a moving target, right? So research is going on right now. It's really hot topic. And the website you might go in, it looks different than what it looked last week. And it did, annoyingly, it did look different last quarter. So if you have something for last quarter, the websites we're using look different. So we had to go in and make some changes to how we presented it, because I get for the CDC, which reorganized the whole tables, right?
CDC changed their website while I was working on it. It updated.
So it was organized this way, and now it's organized that way, the table. So it's a less, you have to scroll a lot to get all the information. But anyway, so so that's the exciting thing, right? It is live. It's not just canned and you have always the same answer. You might find new answers when you go in there than what we already was there 6 weeks ago, right? So and we can't tell you, yes, this is right? So if you email me and say, oh, is my video okay? I don't know. I would have to do all the research you just did and cross-check, right? I don't have the right answer here. It's an open-ended thing. So do your thing, do your best, what you learned, use all the website, and have fun with it. Discuss with your groups. That's your research group, right? Your research group is your group you can bounce off ideas. And so depending on what group you are, you get one of these vaccine, and then we just let you loose and put together your video, submit it by Monday next week. And so 6 to 8 minutes. Remember, you're meeting for 1 hour, 20 minutes, right? That's what you're meeting for. So it's 80 minutes. So you have 8 groups. If you all make 12-minute videos, you can't even get through, right? You also want to take notes, watching the videos of your peers and evaluate them we have a rubric for that. Let's see, so this is a submission. So if you go to group presentations, there's a submission link here where you're going to submit your video. So you might work on a narrated PowerPoint, but then if you do that, convert it into a video, because your TA has to quickly show these, so they can't fumble around, make it work, right? So if you do a narrated PowerPoint, convert it to a video. There's a save as video or something like that. Also, the video should not be 40 gigabyte, right? You have HD, 4K, you're gonna break the system. So make sure if it's really big and it doesn't upload, and if you're not really tech savvy, but you just have to look at the size of your video, right? And if it's more than 1 gigabyte, then we have a problem. And only one of you needs to submit a video for your group. And then remember, it's really important to put your section number and your group number. So it's one big packet of all these videos, and we probably have, I don't know how many sections, how many groups, 8 times. No, like we have 200 times divided by 3, right? It's about 70 videos in one folder, and your TA needs to quickly find your video. Otherwise, they spend 20 minutes looking for your video, right? So this seems silly, but it's important when your TA is in section with you and can't find your video. And then you start scrambling, and then you can't show the video. Okay, so this is important. And then the lab activity is you're going to be evaluating your peers, right, while you're watching the videos. I don't know whether I have a slide for that. I might not have a slide for that. No, I don't. So that's, I guess that, oh, I show that next week, right? So we have another lecture next week, and I already prepared it while thinking, right? So I'm way ahead. So next week, I'm going to show you, there's a rubric, and you're going to evaluate your peers. Right now, just focus on making the video and making it at under 10 minutes, meeting with your group, discussing, you know, your vaccine, do some research. You know, you can do it all at the same time, or you do the research before and then meet with your group and quickly go through what you found and discuss how you want to organize the presentation, and then make a date to actually record the presentation, right? So that's what you're going to want to do for this week, so you can submit your video. And then next week, Monday, I'm going to talk about what's going to happen at the end of the week with that. And so here's my last slide, just a summary of all the reminders, right? So before tomorrow and Wednesday, remember to do the labsters, videos. When there's a lot of videos, PCR, familiarize you with PCR. 5' 3' direction is very important. You know, Google it, or you know, if you watch my lectures and you're still confused, Google it. It's a very important thing. Also, how you order primers, just as a hint there. Take the pre-lab quiz, right? Then on Friday, the assessment for your second writing assignment, it can't be extended. If you miss it, you miss 25 points. It's a higher stakes this time, right? And then the optional reflections open, and then due on Monday at 5 p.m. And then also Monday, 5 p.m. is the group video project. And that's it. So I have 7 minutes surprisingly left. So we could go through some questions if anybody has questions. We are open for answering them. Is there anything, team?
There's nothing in the chat that hasn't been addressed. I have gotten a couple of private questions about the Turnitin scores, and those aren't visible yet. So it usually takes Turnitin a couple days to process those plagiarism scores. So they won't be available until a little later in the week.
Yeah, and so it's like the last time. So we are surprised as you are. You know, I'm just waiting for them. I think 2 weeks ago, they came through on Wednesday. And then once I had them, I did send out all the notifications to all of you, the personal notifications. So that so once I have them, I send these out, and then you start responding to me, and then I'm busy all the rest of the week just going back and forth with people who are concerned. So you already went through that once. So so maybe I can explain again. So if you have 10%, not every 10% is the same. If all your citations match and one sentence and it's 10%, then you know that might be accidental, or some of your citations match. But if you have 10% and it's a little bit of the title, a little bit here, a little bit there, and it's throughout the whole thing, that 10% is a different 10%. I see you use somebody else's paper. If I at the end of the quarter have 3 papers of you and you use the same paper of somebody else and are getting the same name, right? it's really clear you use that person's paper. So then that's a whole different situation than randomly matching here and there, you know, a sentence. So, and I'm trying to explain that when I meet with you, but don't be scared. It's also a moment where we can learn what is plagiarism and what you shouldn't be doing. And hopefully most of you started to understand that. But, you know, if you got a warning and then at the end of the quarter, you just run out of time and it's 40%, I do have to report you. So, but then also again, don't stress too much. Sometimes you have random matches and that happens, but we basically have to look at it case by case. So if you had 0 and now you have 10 or 15 or 20 or 40 in your second, rewatch the plagiarism tutorial and the videos in there to try to understand what happened before you come to me. So what sometimes happens is people go into denial. They say, oh yeah, it can't be me, it's just random. And then they watch it and then, oh, now I know. I didn't know. I had no idea that that would be, but now I know. But you know, your instinct reaction might be, no, it's not, right? So do the work before you, so we're on the same page if we are meeting. And don't be afraid of me, I don't bite. We're just gonna have a conversation. All right, so then I can let you go and have a... Yeah.
Andre, do you have your hand raised? If you have a question, you're free to unmute yourself and ask the question.
Hi, thank you. When I was reading the peer review for the second scientific writing assignment, I noticed that the texts that were provided to us, they didn't cite every sentence. Did we have to do that for our second writing assignment?
So they did cite every sentence or they didn't cite every sentence?
They did not cite every sentence, like in the 2 example papers given.
So how you cite is very context dependent. And I'm doing the writing stuff this session, so I'm reviewing these all the time. But the context dependent aspect of it is, if you're talking about like a study and you're doing multiple sentences that are about the same study from the same source, you don't need to cite every sentence. So if it's very clear to your reader that the information you're providing is all coming from the same place, citing after every sentence is kind of redundant. If you're giving like a bunch of little bits of background information, they're coming from different places, or if maybe they are coming from the same place, but it's really not clear reading the paper, then you might want to cite after every sentence. But like often when you're doing a few sentences about one study, there's no reason to cite for every sentence. Those are real student examples. So sometimes a student who's really nervous about being accused of not citing enough will cite after every sentence just to be safe, but it's definitely not necessary if the context makes it clear that you're citing from the same source. Does that make sense?
Yeah, thank you so much. Also, would we put the citation at the first sentence where we mention the study or the last sentence?
That's a good question. I would generally put it after the last, but I don't actually know if there's any like, I don't know if there's a technically correct answer to that. It makes the most sense to me usually to put it last, because then it's like, okay, this whole block came from that. But Dr. Flugel, do you have like a preference on that?
Well, yeah, I mean, it's depending what Rachel's on context, right? So if you read and then at the very end is one citation, and you think, well, where's this all other stuff coming from, and you're not sure, then maybe it's missing, right? But if it's really clear, it's one sentence, this and this and this and this, and then there's like one citation, it's clear to the reader, right? So it's depending on how it's written. And then just to note, the examples, the practicing peer review, they're not good examples. They're just for you to evaluate what's right. They're not like, oh, here, this is our best example how to write a paper. They're not. They're just like some 2 students' paper we just pulled. They're not expert papers. And then you have to evaluate and agree. And I guess we evaluated them to say, well, we think it's this and this and this, but it's not, by no means takes that and say, okay, this is going to be my paper because this is a perfect written paper. It's not.
All right, thank you so much.
All right. Well, good luck, everybody. It's a big lab. I hope you're all excited about SARS-CoV-2, and I see you next week for the last time.